like, I feel like
And I literally so mad. I have to say you woke us up.
okay, wait, yeah. This a 3 or 4 version of it.
Yes.
Wait, yeah.
That class got me. That was hard. You could have just sent email.
Turning out so high. Wait, it's not they're any more free and able to do stuff. Like I stack all my classes. I have to. It's like, wait, yeah, everyone.
We are spending a little bit of time on nucleic acids, their structure, and also the energetics involved in synthesizing nucleic acid. And then talking about a couple of experiments that prove that DNA is genetic material and not proteins, which is thought to be the case historically. So getting into this. So this is the structure of RNA strand in a DNA strand. Very similar in terms that the nucleotides are held together with what are called phosphodiester bonds, linkages here between neighboring nucleotides. Main differences between DNA and RNA are going to be the sugar structure. We have ribose sugars. DNA has deoxyribose. This H replaces the hydroxyl group that is present in ribose sugars. And this is at what we call the 2' position. A ribose sugar is labeled, its carbons are labeled by numbers. And this is 1, 2, 3, 4. So a ribose is a 5-carbon sugar. At the 2' position, they differ between DNA and RNA. And this impacts the stability of RNA because this hydroxyl group can be reactive. And we'll talk about that in the next set of slides. Meaning, RNA can be catalytically active. It can actually facilitate enzymatic reactions. And it was one of, evolutionarily, it was thought that RNA actually was the first molecule to catalyze reaction. Okay, other things to point out. The phosphate group in the backbone, this oxygen can be protonated, but its pKa is about 1. So super acidic. And so at all physiological conditions, it is deprotonated and it has a negative charge. So the phosphate backbone of both DNA and RNA is very negatively charged in the cell. And so if you have proteins that bind DNA or RNA, DNA binding proteins, RNA binding proteins, they tend to have positive charges on them where they sit down on this structure. So, which potentially are composed of amino acids, side chains that are positively charged. Okay. So, and that's why it's called nucleic acid. It's very acidic in this one. All right, so the bases here, you should be able to identify these by sight. Understand which ones are purines, that's the adenine and guanine, and be able to identify which one is which. And then the pyrimidines, these three major ones, you don't have to know the modified base of methyl cytosine, but you should know cytosine, thymine, and uracil, be able to identify those and know that those are pyrimidines. So of course, in DNA, we have A, G, C, and T, and in RNA, instead of T, we have U. Which two bases tend to interact with each other if in a double-stranded piece of DNA? What are our base pairs? Yeah.
A and T and C and G.
Yeah, A and T and C and G are base pairs. And when you form a base pair, what type of bonding is that?
The hydrogen.
Yeah, those are hydrogen bonds. So the hydrogen bonds we talked about. C's and G's are able to form 3 hydrogen bonds between them, and A's and T's can form 2 hydrogen bonds. And that affects the chemistry of CG-AT base pairs. C and G base pairs are easier to melt apart than A's and T's. So we'll talk about that later as well. Okay, the terminology of nucleic acids. You have a structure like this that has the ribose, the base, and the phosphate. That's called a nucleotide. Sort of the typical structure we see there. In this case, it's a nucleotide monophosphate because there's a single phosphate on the end. Last week, we talked about a nucleotide triphosphate. That was ATP, right? And so that would have 3 phosphates on the end. This is a monophosphate in this structure, and that's called a nucleotide. And just to differentiate between that and another term you might hear, which is a nucleoside, same structure with the base and a sugar, but it doesn't have a phosphate on that. So that's what a nucleoside means. You hear that term. Okay. And then you can differentiate between adenosine and deoxyadenosine, or guanosine and deoxy. This is the RNA version. This is the DNA version, based on if it has the hydroxyl group here, or all of these have the hydroxyl group, or if it has just a hydrogen, that's deoxy and that's DNA. Okay, questions on any of that? Okay, so if we are working in a lab, sometimes we need to quantitate how much DNA or RNA we have. And you can use a machine called a spectrophotometer, which shines a wavelength of light through a sample, typically in a cuvette, which is transparent. So light can go through it, and then a certain amount of light would be absorbed by your sample, and a certain amount is transmitted through your sample. That quantity is red. But the amount that is absorbed is going to be relative to the amount of nucleic acids in your sample if you shine a specific wavelength of light. So RNA and DNA, these are all the versions. This is all RNA, but T and nucleotide thymine monophosphate also absorbs light. Maximally at 260 nanometers is where you can measure DNA and RNA. So it cannot tell if you put a sample in the spectrophotometer. It will not tell you if it's DNA or RNA, but it will tell you how much light gets absorbed by your sample, and then you can quantitate how much nucleic acid is in there. Has anyone ever done that in the lab to quantitate DNA or RNA? I'm just making nanograms now. But you will, if you do this, sometimes you will also get a reading. You will also shine a different wavelength of light at 280, and you get a ratio. That's a 260-280 ratio. What is being measured at 280 are proteins. So if you have a 260-280 ratio, that will tell you how much nucleic acid to proteins you have, and that will tell you if your sample is contaminated with protein. Ideally, you want DNA or RNA that's pretty pure if you're running an experiment with it. And so we'll talk more later about why amino acids and proteins absorb light at 280. But those are the two numbers you might think. Questions on that? All right, so our RNA structure here, like I said, it of course transmits genetic information from DNA and our cells that contain all the sequences of our genes. Yeah.
Sorry, so the amount absorbed is relative to what?
The amount of light absorbed is relative to the concentration of nucleic acid in your, so yeah, the more concentrated your DNA sample, the more light it will absorb. And it's absorbing it because the bases, these conjugated double bonds absorb light. So that's what's actually absorbing the light. Okay, so our RNA, as we know, is sort of a conduit between DNA, which contains all of our genetic material, and the protein translation machine, right? So if we want, most of the time, our cells are not expressing all of the genes that we have. So we can sort of pick and choose which genes are expressed by only transcribing certain RNAs and passing along that information to the ribosome. So RNA transmits and stores genetic information, but it can also undergo catalysis. And that is because of this reactive group, which is different from DNA. So how does this work? If we have a strand of RNA like this, what can happen is this hydroxyl group can become reactive, meaning it wants to form bonds with other atoms nearby. One way that can happen is if you have a little bit of base in your solution where the RNA is. The base will tend to pull the proton off of this hydroxyl group. That makes water, but it also makes this oxygen reactive. It has a negative charge and it wants to form a bond with a neighboring atom. And so quite often, because this phosphate is nearby, it will form a bond with that phosphate. Okay. What happens then is that this phosphate can only have so many bonds. And so it drops the bond with the hydroxyl group in the neighboring nucleotide. And so now that RNA is cut in two pieces at that location. So a little bit of base makes this quite reactive and you end up cleaving, or the RNA self-cleaves itself. So if anyone has ever worked with RNA in a lab, this process of sort of self-cleaving can happen quite easily, not just by adding base. Anything that's going to deprotonate this can make it react. So you have to be very careful when working with RNA so it doesn't get degraded. DNA, of course, has only a hydrogen here. So it's less reactive. It doesn't self-cleave, much more stable. And DNA tends to be in double-stranded form. So it's sort of extra protective. So once DNA evolved evolutionarily, it's much more stable. It could grow longer and encode more genetic information. And so we started to have longer genomes once DNA evolved. Okay, so thinking about the energetic, bringing back our idea of energetics and thinking about how we can synthesize long strands of DNA or RNA, kind of go at this and think about this process of synthesis. So what sorts of enzymes are synthesizing DNA and RNA in this cell? What are those called? Remember? Yeah.
Transcriptase?
Polymerase. Yeah, but yeah, a transcriptase would be could be transcribing as well. Yeah, typically, it's another term for it. A DNA polymerase, an RNA polymerase, catalyzing addition of nucleotide onto a growing chain. That's basically what's going on. Okay, so that's the enzyme that's doing that. But what are the energetics of that reaction? Well, we see a chain of nucleotides. We basically see nucleotides that have a single phosphate, a ribose, and a base, right? So it looks like nucleotide monophosphates in a big chain, right? If we think about growing chains in that way, we can sort of look at this middle reaction in this description there. If you had an existing poly chain, polynucleotide chain, and if we tried to add on another nucleotide monophosphate, any base, doesn't really matter, this N just means anyone, and that's a dehydration reaction. Is adding a nucleotide monophosphate onto an existing chain favorable? No, not favorable to do that. Okay, so how does that happen? How do we have a chain with an extra nucleotide monophosphate? The way that works is that in the site of the polymerase, we don't put in a nucleotide monophosphate. That's not one of the reactants. The reactants is this. What is this? Some nucleotide, some base, but it has a triphosphate. And we learned about the energetics of these molecules last time, right? Is it favorable to remove one or two of these phosphates? Yes, very favorable. So that's great because that can be the reactant inside the polymerase rather than a nucleotide monophosphate. Okay, so you have an existing chain. The polymerase is trying to add on a new nucleotide here. The other reactant is going to be one of these. What happens is that hydroxyl group will form a bond with this phosphate, and these two phosphates are released. So that releases a lot of energy. Okay. If we had a nucleotide triphosphate and you add water essentially, and you remove these two phosphates, which are called pyrophosphate, that's the abbreviation, pyrophosphate. Is that favorable? Yeah, so removing these two phosphates from a triphosphate, very favorable. So we're coupling those two inside the enzyme's active site, the DNA polymerase RNA. And then additionally, even if you add those two together at standard conditions, that's favorable. But additionally, what sort of happens after the fact is this pyrophosphate is further hydrolyzed by water. So once it's removed, it's also attacked by water and just cleaves into two separate phosphates. Also favorable. Okay, so then we can sort of add all these up, and the net reaction is to have an existing polynucleotide chain, provide a nucleotide triphosphate, have water, and we can create a bond that has an additional nucleotide on it, and we're left with two individual phosphates at the end. And that's very exergonic. Okay, so that's how you can make a reaction. It's a building reaction that on the surface is an unfavorable reaction. You can make it favorable. Questions on those steps or any of the parts of that reaction? Right. So this is a look at our chains of nucleotides. This is sort of on its side here. And just to demonstrate that the covalent bonds between neighboring nucleotides are going to be between the ribose and the phosphate, the ribose and the phosphate, right? That forms the backbone. That's sort of what that term is for a piece of RNA or DNA. the bases kind of stick off the other side here. And at the 5' end, you would have phosphate that is just hanging off the end, and then at the other end, you would have a hydroxyl group. Whenever you say a sequence of DNA or RNA, the sequence is always written from the 5' end to the 3' end. That's just by convention. That's how we say a sequence. So it would be, if this is the 5' end, you would say this base, and this base. CGT. What happened? Okay, so looking at these two experiments here that are, and there's also in your problem set, there's a reference to another set of experiments that also help to prove that DNA was the genetic material. So all of these experiments together help to prove that DNA was the genetic material of non-protein. So I'm just going over two of them, but there were others that contributed to that knowledge, just so you're aware of that. So this one was done in 1944 by Avery, McLeod, and McCarty. And this concept was called transformation, what they showed here. And the idea is that you could change the characteristic or a phenotype of an organism. In this case, we're talking bacterial cells, by transferring pieces of DNA that encoded those characteristics. And that's called the transformation or transforming principle. Okay, so the idea here is that they had two populations of bacterial cells. One was pathogenic, meaning it caused disease in mice. And it had a cell wall that was smooth in terms of its look. Microscopically, you can see it had a smooth cell wall. They also had a population of cells that were non-pathogenic, not disease-causing, and they had sort of a rough cell wall. So the question was, could we transform the non-pathogenic cells by providing them with DNA from the pathogenic cell? And the way they did this experiment was to heat kill these cells. So they would break them open and the DNA gets released. These cells are now dead. But you mix the contents of these cells with the non-pathogenic cells. What happens in a culture like that are bacterial cells will take up some of the sample from their environment. And in this case, some portion of these cells took up DNA from the environment and from the cells that were killed here. And then some percentage happened to take the genes that encoded the pathogenic characteristic and also the smooth cell wall characteristic. So they allowed this mixing to occur. Some of the cells took up this fragment. They allowed this population to grow and divide. Some percentage happened to take up the gene that encoded pathogenicity and also the smooth cell coat. And so they changed their phenotype or their characteristic. They became pathogenic and they had a smooth cell coat. So the concept here is that whatever was released from this host cell was able to survive heat, which most proteins cannot, and it was able to change the genetics of this cell and change its characteristics. So that was a set of experiments that indicated that it was likely DNA that encoded genetic characteristics. The second experiment that we'll talk about is called the Hershey-Chase experiment. This is also a classic experiment, and this was done using bacterial viruses called bacteriophage and their host cells, which are bacterial cells. This The way that phage work is they have a protein coat. They're very simple protein coat, and then it that protects and houses the DNA, which is the genetic material of the phage. The way infection work is that the phage will sit down on a host cell, it injects its DNA to initiate infection. So the experimenters wanted to determine if the production of new phage was mediated by the DNA, which was injected into the cell, or was it mediated by the protein coat that was hanging out there on the outside? So what they did was to use a lot of radioactive isotopes, and they grew the phage in P32, phosphate that was radioactively labeled phosphate. Phosphate is going to label the DNA, right? Because DNA contains phosphate molecules. So P32 labels the DNA. They also labeled the protein coat with S35. So sulfur is in some amino acids, but it is not in DNA. So you can label those with two different isotopes and label the phage in different ways. Okay. So we have these radioactive phage. They allow them to mix with E. coli, which is the host cell. The DNA gets injected. They then separated the protein coat from the DNA that was injected into the host cell. So you used a mixer and they got rid of these protein coats. And then the question was, can the DNA that was injected here, is that sufficient to direct the formation of new phage particles? Does it have the genetic information to make new phage? And the answer was yes. The DNA was basically replicated and transcribed and translated by the host cell to produce viral proteins, which makes all the new phage particles, and new genomes that get packaged. So the resultant progeny phage there do not have any S35 because that sort of empty protein coat was removed, but some of the DNA was still labeled via the radioactive nucleotides that were in the DNA that was present in the host cell. So that was sort of the final experiment that indicated that DNA was sufficient and able to produce new phage. The protein was basically just there as a delivery. Questions on that experiment or the other experiment? Yeah.
Do phage just like grow out of the live cell, or just like
This typically they will lyse the cell, so they break it open and then they burst out. Yeah, so it ends up typically killing the host cell. Mm-hmm. All right. Let's go ahead and do our problem set.